we're switching gears. The earlier talk that I gave was obviously on the adult life stage, and so kind of continuing on this theme that Corey started of looking at the younger life stages. And this work is also part of that Together with Gigognac project that I mentioned earlier. So it's done in collaboration with Saugeen Ojibwe Nation, Fathom 5 National Marine Park, and then the Upper Great Lakes Management Unit, and then my team in aquatic research. And also some of the work is funded through COA. And then I wanted to give a special thanks to Isaac Hebert, who's a biologist working in my team, who is really the person doing a lot of the analysis of our larval fish data. So he's put in a great effort for this work. So I'm super appreciative of Isaac for everything he's done on this work. So I'm going to talk today about both lake whitefish and Cisco. Like at many other lake committee meetings, most of my work and most of what I've focused on has been lake whitefish and trying to understand how the ecosystem changes, including the reduction in productivity, has affected the larval life stage of whitefish and how that's influenced recruitment. But for today's talk, I'm actually going to talk about Cisco as well, and this is really inspired by work that other people have been doing. So if folks were here yesterday during the Lake Michigan session, Scott McNaught gave a really great talk about some of the work they've been doing to try to understand differences between whitefish and Cisco at the larval stage. And then also Taylor Brown has been doing some work also looking at Cisco and whitefish. So you know, a lot of my program is focused on whitefish, and now we're just starting to turn our attention to try to look at Cisco as well. So obviously both of these fish species are very important in Lake Huron. So lake whitefish is incredibly important in our commercial fisheries, both indigenous and non-indigenous fisheries. And Cisco is a very important pelagic prey fish within the Lake Huron ecosystem. It's our native prey fish for our top predators. And you know, at the juvenile and adult life stage, these two crigonines have very different life histories and ecologies. So, you know, lake whitefish are, you know, a large-bodied benthic species. So they're living on the bottom mostly. They're eating benthic invertebrates and some benthic fishes, whereas Cisco are primarily a pelagic planktivore during the juvenile and adult life stage, although they can eat fish as well. However, during the early portion of their life cycle, they actually are very similar. So there is some overlap in their spawning habitat, and as Corey nicely sort of went through, you know, what happens when these fish hatch and swim up the surface, well, lake whitefish and Cisco are very similar during this time. They're both planktivores. You can't even tell them apart visually. You have to use genetic identification to be able to tell them apart. So the work that I'm going to be presenting is trying to understand how processes affecting the larval life stage go on to influence population dynamics. And kind of the backdrop to all of this, of course, is the ecosystem changes brought about from Dreisseniid mussels and that reduction in productivity, which has led, you know, just due to their filter feeding capacity and their influence on like nutrients. We've seen reductions in zooplankton populations throughout Lake Huron, and this is what we believe is contributing to reduced growth and poor survival of larval lake whitefish. However, what's really interesting about these two crigonines is that despite these similarities early in life, they've shown different sort of trajectories. So we've heard about the whitefish declines, of course, that have occurred in Lake Huron, but what's interesting is that Cisco have actually shown some opposite trends. So we're starting to see signs of recovery or increases in population abundance of Cisco. So the graph on the right here is showing some recovery in the main basin. This is from M&R offshore index netting. We heard Tim O'Brien earlier talking about trends in Cisco. So their acoustic work is also showing this increase in the main basin of Cisco, as well as in the North Channel. So it kind of begs the question, well, if you know, Dreisseniid mussels are causing reduced zooplankton and this is affecting recruitment of whitefish, like what's happening with Cisco? Why do we see the opposite trend? And so I've just recently begun to look at the larval Cisco data that I have, because while I was out sampling for whitefish, we were catching lots of Cisco. So this talk is a little bit exploratory, starting to look at some of the differences and similarities that we see at the larval life stage between these two species. So for this work, what we do is we go out in the springtime during the hatching period for these fish, and we're doing surface tows with a larval fish net at the same time that we're doing tows with a plankton net. So we're sampling both the larvae and the plankton, and the idea there is that we want to look at, how many larval fish are there, their growth rates, their diets, all of that good stuff, as well as looking at the food that's available in the environment at that time for these fish. And so we basically go out every day that we can after ice out and do our sampling, and this is so that we can really capture that full period of that larval period, because what you see is like the fish hatch, the numbers kind of increase, and then they taper off again. So we're really interested in characterizing the production of these larval fish. And so our sampling has been done at a number of different areas. Most of our work has focused on the Fishing Islands spawning shoal complex in the main basin. Here, this is a very important spawning shoal to whitefish, and I will show in a moment to Cisco as well. And fortunately, we have time series actually going back to the 70s and 80s before dry-scented mussels were in the system. So it's offered a really nice time series for us to look at. We also have sampling done in southern Georgian Bay, up in the North Channel, and a little bit north of the Fishing Islands in the central main basin. So some of the questions that I'm exploring today and that I'm interested in looking at with this data set is, are there differences in the ecology and dynamics of Cisco and lake whitefish during this larval life stage? So some of the things that I'm looking at are spatial and temporal patterns. So are there differences among the basins in the numbers of these larval fish produced and in species composition? What are the trends through time, given the trends that we're seeing at the population abundance level? And then also, is there overlap in distribution during the spawning period between these fish? Looking at differences in growth, and then also differences in the timing of hatching, which could influence whether there's a match or mismatch between when these fish are hatching and how much food is available for them in the environment. And then also we're looking at differences in diet. I'm not going to show any of that data. We're still working on it, although we're already starting to see some really cool things. So if there's time at the end, maybe I'll talk about that. So overall, basically what we're just interested in looking at is whether any of these differences that we see at the larval life stage, could they help explain the different trends that we're seeing in population abundance? So that decline in whitefish, but that increase in Cisco. Okay, so this is showing the proportion of our larval fish by species for these different areas. So we see very different patterns around the lake. So up in the North Channel, most of the larval fish that we're capturing are Cisco. And so, this kind of speaks a little bit to what Corey was saying, where they weren't capturing a lot of larval whitefish up there. And then you see that down in southern Georgian Bay, down here, we're catching all lake whitefish, so no Cisco to be found there. Then we see in the central main basin, our two sites are kind of a little bit more variable, where mostly Cisco, but a few more whitefish in the mix as well. Okay, this is showing the larval densities. So this is focused on lake whitefish for our different locations for different years during this contemporary time period that we're focused on here. So obviously, the obvious difference here is that Fishing Islands, we're catching far more larval lake whitefish than the other areas. So up in the North Channel, we're capturing very few lake whitefish. We see more at Fathom 5 in the central main basin, and not very many in Georgian Bay. For Cisco, somewhat similar in that the Fishing Islands are also producing a lot of larval Cisco, relatively speaking, but we do see more Cisco in the North Channel and at Fathom 5 than we did for whitefish. And you will note there is still substantial year-to-year variability, which is basically saying that, There's other factors at play here, like, for example, climate potentially, how quickly the water is warming, that is going to influence that year-to-year variability. Okay, this is showing the distribution of larvae that were captured for whitefish and cisco. This is the fishing islands spawning shoal complex here. It's a very large area, but a couple of take-home messages. Larval lake whitefish tend to be more clumped, and they're found in more of the sort of the embayments, so kind of closer to shore. Cisco have kind of a more broad or spread out pattern, and we see them more predominantly than whitefish in the offshore areas. So while there is overlap in this larval habitat, there still are differences. Cisco are found a little deeper, and when you look at catches by depth, you see that reflected as well. Okay, so in terms of growth rates, I find this really cool. So basically, lake whitefish are starting life larger. However, cisco grow faster. So by the end of this larval period, the cisco have more or less caught up to where those lake whitefish are. And Corey was talking about the differences in growth rates that he was seeing for whitefish across the different regions of Lake Huron. Some of our stuff does reflect what he's seeing. So for example, in the North Channel, we actually see faster growth rates for our larval cisco and whitefish, but slower growth rates in the central main basin in the fishing islands. Hard to see based on this, but that pattern is there. So this is just looking at the correspondence between cisco growth and whitefish growth for our larvae. And this is for showing all of the data for different years and also the different sites, all shown together. But the basic take-home message here is that when cisco are growing well, whitefish are growing well as well. Sorry, that didn't sound very good, but anyway, they're both comparable in terms of their growth. So this is despite the fact that cisco are still growing relatively quicker than whitefish, but we still see this pattern. When cisco are doing well, whitefish are doing well. Okay, so this graph now is quite interesting. So this is showing now the comparison between our more recent data, that's the contemporary portion of the graphs, with that historical time series that I mentioned from the 70s and 80s. And so what we see here, and this is published in a paper if you're interested in looking at this in closer detail, but what we see here is that for our larval lake whitefish, we kind of have low densities of fish early on. Then we see this ramping up where larval numbers are increasing, and this really closely reflects the population trajectory of whitefish. Then in the contemporary time period, we see a reduction of up to about, I think it's like 75% reduction in larval production, although we still see some year-to-year variation. Now we're going to look at our cisco, and here we see the opposite pattern. So here, historically, cisco larvae densities were much lower than they are now. So we're seeing those higher catches of cisco. So this does reflect to some extent what we're seeing in the population abundance that I showed at the very early part of this presentation, where whitefish are declining and cisco are increasing. Okay, and then this is going to be really hard to read, but I wanted to put it up anyway. And this is just looking at the correspondence between zooplankton densities for these different areas and the numbers of larval fish. So zooplankton densities are along the top, and then we have lake whitefish larval densities in the middle, and then cisco densities along the bottom. And so this is showing the patterns for the different sites. So North Channel, Central Main Basin sites, which are Fathom 5 and Fishing Islands, and then Georgian Bay. Each color, which you don't really have to try to look at, are different years. And then so this is showing the trends by day of the year. I will note, I'm changing the axes here to kind of highlight the differences. But one thing we do clearly see in our zooplankton data is that we do see higher zooplankton densities in the North Channel and very low zooplankton densities in the Central Main Basin. However, the purpose of looking at this is to look at whether there's any differences in the timing of when cisco and whitefish larvae are hatching. Again, to get at this idea, like, is there a mismatch between when these fish are hatching and how much food is out there for them? And so the take-home message here is that really they're very similar in when they're hatching. So it's not like, you know, whitefish are hatching really early and cisco later or anything like that. But what's interesting that we do see in these data is that there's a much more chance of that mismatch occurring in the central main basin in the fishing islands area, because we see that zooplankton peak is reached, so that zooplankton bloom happens later in the season, whereas in Blind River it's happening much earlier in the season. And in the fishing islands as well, what we're seeing is that there's some years where there is really no zooplankton bloom, and this is kind of corresponding back to what we know about what's happening with phytoplankton populations throughout Lake Huron. And what we've seen from some other studies is that there's been a loss of the spring phytoplankton bloom as a result of the filter feeding capacity of dreisseniid mussels to really filter out the whole water column. And so we're kind of seeing that reflected in the zooplankton data, but more so for that central main basin. Okay, so that's what I have so far. As I mentioned, we are looking into diet differences between cisco and whitefish, and we are seeing some interesting things there, so you can stay tuned for that. This is really kind of a start for me of looking at the two species. But to summarize, we do see large regional variation in the relative proportion and abundance of our larval cisco and larval lake whitefish. We had overlap in their habitat, although relatively cisco were found deeper. Larval hatching timing was similar between the species. And we did see that the trends in Coregonine larvae for the two species did seem to reflect the trends that we're seeing in population abundance, so of the adults. And we saw that the conditions for growth among the species is linked. So, you know, good years for cisco growth are also good years for whitefish growth, which kind of tells me that you know, there's still some similarities in terms of how these larvae are experiencing the ecosystem conditions and that year-to-year variation. So kind of like to wrap it all up, I would say that we see several differences between the two species, but also a few of these similarities in how they're responding to the ecosystem changes. So it's like a little bit of a mixed bag, and I feel like we're sort of just getting to understanding like what's happening and why these two species show such different trends at the population level. And as I mentioned, Scott McNaught gave a presentation yesterday which kind of looked at things like metabolism between larval cisco and whitefish. There's also work being done by Nicole Berry looking at UV and how it differentially affects larval cisco and larval whitefish, and they're finding some really interesting things that also are sort of like helping us with this puzzle of why these two species show such different trends. And with that, I'll say thank you very much and happy to take questions.